Here's some help with identifying dipoles. The question says, show the dipole in each of the following bonds by drawing an arrow. Write delta, that little squiggly looking D is the lowercase Greek letter delta. Write delta positive next to the atom in each bond that has a slight positive charge, and delta minus next to the atom that has a slight negative charge. So here, we're trying to show a separation of charge in a bond. And to understand that how that happens, the first thing we need to do is understand something called electronegativity. Electronegativity is how much an atom attracts valence electrons. So how much do those protons in a nucleus attract the valence electrons in an atom? That increases as you go up on the periodic table, as the valence electrons, the outermost electrons on an atom, are closest to the nucleus, and as you go to the right on the periodic table. That's as you get more protons in the nucleus. So an atom attracts valence electrons more as you go up and to the right on the periodic table. Let's think about the implications that has when you have a bond between two different atoms. So in this case, carbon and oxygen. Now if you look, carbon is here, sort of in the top right, and further to the right is oxygen. And that means that oxygen is more electronegative. So oxygen attracts electrons, valence electrons, more than carbon does. So when they're trying to form a bond and share these two electrons, the oxygen is going to pull those electrons to itself more of the time. Because electrons have a negative charge, and they're spending more time around the oxygen, that oxygen is going to have a slightly negative charge. And that lowercase Greek letter delta, that squiggly D, means slightly. So it's not a full negative charge. The electrons are still being shared between carbon and oxygen, but they're spending more time around the oxygen, making the oxygen slightly negative. So the more electronegative atom gets a slightly negative charge. The other atom in the bond is losing those electrons some of the time. They're not spending as much time around, in this case, the carbon. So it has a slightly positive charge. And so that is a dipole. Di in dipole means two, and the pole means ends. Just like the north and south pole are the two ends of the Earth, dipole means you have two ends on this bond. You have a positive end and a negative end. And it's created by differences in electronegativity. Different atoms attracting the electrons in a bond more or less. So one way to draw dipoles is with this delta plus and delta minus. There's another way using an arrow. The arrow points in the direction of where the electrons are going. They're pointing toward the more electronegative atom, the atom that is attracting the electrons more. Not only do you write the arrow, but you also write a little plus sign on the tail of the arrow to remind you of the slightly positive charge that's on the atom of that side. So these are the two ways of drawing the dipole in this bond. One way using the delta plus and delta minus, the other way using that arrow with the positive tail. Where would you find something like this? What type of a molecule? So one molecule you would find this dipole in is diethyl ether, which was an early anesthetic used in surgeries. There's a picture of the molecule on the bottom right, and right there we have that bond between carbon and oxygen. And just like we saw before, because oxygen is more electronegative, it's going to be slightly negative in this bond, and the carbon is going to be slightly positive. There's another bond between oxygen and carbon, and so here the oxygen again is slightly negative and the carbon is slightly positive. And it's those partial charges. Remember, charge makes things sticky. So it's those partial charges that make this molecule stick to the proteins that make people insensitive to pain and make it work as an anesthetic. It's not used as an anesthetic anymore because diethyl ether is really flammable. Here you can see it burning. Okay, let's try to identify the dipole on this bond, a bond between nitrogen and carbon. So nitrogen is here on the right, carbon is here. Because nitrogen is further to the right, it is more electronegative. That means that it's going to attract the electrons in a bond to it more than carbon will. Because electrons have a negative charge, that nitrogen is going to be slightly negative. And the carbon, which is losing the electrons for more of the time, is going to be slightly positive. So the more electronegative atom gets a slightly negative charge, and the atom it's bonded to becomes slightly positive. You could also draw that with an arrow. The arrow points in the direction of where the electrons are going, toward the more electronegative atom, and the tail of the arrow gets a little plus charge. And that is the dipole in this bond. Where would you have that dipole? One example is the amino acid glycine. So if you think of proteins as chains, long chains, that then fold up to form what we think of as a protein, like our muscles, each link in that chain would be something called an amino acid. And there are 20 different amino acids, but glycine is the simplest one. And in glycine, you have that bond between nitrogen and carbon, and just like we saw, the nitrogen is more electronegative, so it's going to have a slightly negative charge, and the carbon will have a slightly positive charge. And it's those little stickinesses, those slight charges, that are going to make one part of the protein attracted to another, and make it fold up in just the right shape to work in our body. How about the dipole between sulfur and hydrogen? If you had a sulfur and a hydrogen bonded, what would the dipole be on those? So sulfur is here, hydrogen is at the top left. Now here, the trend sort of breaks down. The hydrogen is higher, but the sulfur is further to the right. So which one wins? In a case where two elements are not in the same column or the same row, you would want to use a table of electronegativities. So here on this table, a number is given to every element that says how electronegative it is, quantitatively. The higher the number, the more electronegative. So sulfur here is 2.58, hydrogen is 2.20, because sulfur has a higher number, sulfur is more electronegative. So of these two, sulfur is the more electronegative atom. Now because of that, sulfur is going to have a slightly negative charge, and hydrogen will have a slightly positive charge. Another way of drawing that would be to have an arrow going toward the sulfur, and the direction of where the electrons are going, and to have a slight positive charge on the other atom. So that is how you would draw the dipole on that bond. Where would you find this bond? In another of the amino acids, the links that come together to form proteins. There are 20 different amino acids. Another one is cysteine, and this has that sulfur to hydrogen bond there. The sulfur is more electronegative, so it gets a slight negative charge, and the atom it's bonded to, hydrogen, has a slight positive charge. Those charges make two cysteines in a protein stick together a lot. Ultimately, it's this dipole that's responsible for that. And the more cysteines you have, the more they can stick together, and the harder a protein gets. So hair might not have that many cysteines, but as you get to a nail, which is harder, it has more cysteines that are sticking together because of those dipoles. And then as it gets even harder into a hoof, you'd have even more cysteines. So the dipole is ultimately responsible for the hardness of a protein. How about between iron and nitrogen? So iron is here, nitrogen is up in the top right. Because nitrogen is both higher up and more to the right, it is the more electronegative element. So in this bond, the nitrogen will have a slightly negative charge, and the iron will have a slightly positive charge. And you can also draw that as an arrow going toward the more electronegative atom with a plus sign on the tail of that arrow. And that would be how you would draw the dipole on that bond between iron and nitrogen. Where would you find such a bond? On all of your red blood cells. So your red blood cells are used to carry oxygen throughout your body. And how do they do that? There's a protein called hemoglobin that's in them, and on this hemoglobin protein, there's a little group of atoms that looks like this. And on each of these bonds, just like we saw, the nitrogen is slightly negative and the iron is slightly positive. So on each one, the iron is slightly negative. And so iron is really slightly positive here. And it's that slight positive charge that attracts the electrons on a molecule of oxygen. And that makes the oxygen stick to the red blood cell and allows the red blood cell to carry it around your body. Alright, let's try it with a few more. How about with the bond between oxygen and hydrogen? So, oxygen is there, hydrogen is in the top left. Now, hydrogen is higher up, but oxygen is further to the right, so you'd want to consult a table of electronegativities. Oxygen has a value of 3.44, hydrogen of 2.2. Because oxygen has a higher number, it's more electronegative, and so it is the more electronegative atom. The more electronegative atom attracts electrons to itself and becomes slightly negative, and the atom it's bonded to loses those electrons and becomes slightly positive. And another way you can draw that dipole is to have an arrow pointing in the direction toward the more electronegative atom with a little plus sign on its tail. And that would be how you'd draw the dipole on that bond. That dipole most famously exists on water. So you have an oxygen to hydrogen bond here. The oxygen is going to be slightly negative and the hydrogen slightly positive. And on the other bond, the oxygen slightly negative, the hydrogen slightly positive. And that makes water really, really sticky. Every molecule in water, every atom in a water molecule has a charge. And so water is a really sticky molecule. Because it's so sticky, it's able to dissolve so many different things. When you put a lot of different salts into water, different molecules into water, they dissolve because the sticky water pulls them all apart. As you can see here. And the stickiness of water is also, because water molecules stick so much to themselves, it's really hard to rip them apart. And that's why water can absorb so much heat before it evaporates, and is a good way to cool people down in the form of sweat. How about between nitrogen and hydrogen? What's the dipole there? So, nitrogen is on the right there, hydrogen is in the top left. Now, hydrogen is further up, but hydrogen's further to the right. So to decide between those two, because they're not in the same column or row, you would want to go to a table of electronegativities and compare the numbers. So nitrogen has 3.04, hydrogen has 2.2. Because nitrogen has a higher number, it's more electronegative. So of these two, nitrogen is the more electronegative atom. The more electronegative atom attracts electrons more. So the nitrogen will be slightly negative, and the hydrogen that it's bonded to will be slightly positive. Another way to draw that dipole is to have an arrow pointing toward the more electronegative atom with a little positive sign on its tail. And that is the dipole between the atoms in that bond. Where would you see such a dipole? If you go to the hippocampus in your brain, which is the red part here, and then you were to take a slide of those, you would see the neurons. Here, each of those colored balls in the center are different neurons, and you can see all of the sort of telephone wires that are branching off of them, called axons. So, each neuron, if you follow the axon from it, the telephone wire from one, it's going to another so that it can communicate with it. And if you follow it straight to the end, you'll see what's called a synapse, where the two neurons are connecting and communicating with each other. And so what happens in between the two neurons at the end of those telephone wires? How are they communicating? One of the molecules that they're sending across that little gap is dopamine. And you can see that in dopamine, there is a nitrogen to hydrogen bond. That nitrogen, just as we saw a moment ago, has a slightly negative charge. The hydrogen has a slightly positive charge. And on that other one, the nitrogen, again, slightly negative charge. The hydrogen, a slightly positive charge. And that stickiness on those atoms is part of what makes this molecule, this neurotransmitter, stick as a messenger on one end of a neuron to communicate a message. How about between phosphorus and oxygen? So phosphorus is here, oxygen is there, because oxygen is both higher up and more to the right. It is the more electronegative atom. And so in this bond, the oxygen is going to be slightly negative and the phosphorus will be slightly positive. Another way to draw that dipole is as an arrow pointing in the direction of the more electronegative atom with a positive sign on the tail. And so that is how you would draw the dipole in that bond. Where would you see such a bond? A lot of places, but in the most famous place, perhaps, is ATP. So if you notice in ATP, you have lots of bonds between oxygen and phosphorus. And in each of those bonds, the oxygen is slightly negative and the phosphorus is slightly positive. So again, phosphorus slightly positive, the oxygen slightly negative. Now ultimately, it's those charges that make these phosphoruses really sticky, and they end up sticking onto enzymes as a phosphate. So that PO4 breaks off of the ATP and sticks onto an enzyme because of these partial charges. And when this phosphate sticks onto an enzyme, it either makes the enzyme work or stop working by pushing it and changing its shape just a little bit. When it sticks to it, it changes the shape of the enzyme. And so that's how ATP either turns enzymes on or off and makes reactions either happen or stop happening in a cell. And so it's thought of as a sort of energy molecule because it can either get things done or prevent things from getting done. But it all comes down to these partial charges, these dipoles. How about between hydrogen and chlorine? So hydrogen is in the top left, chlorine is far to the right. Because hydrogen is further up, but chlorine is further to the right, you'd want to consult an electronegativity table to be sure about which one is more electronegative. Hydrogen is 2.2, chlorine is 3.16. Because chlorine has a higher number, it is more electronegative, and so of these two, chlorine will be the more electronegative element. The more electronegative atom attracts the electrons to it more, and so becomes slightly negative. The other atom in the bond is losing those electrons, so it's slightly positive. Another way to draw that dipole is to have an arrow pointing toward the more electronegative atom with a positive charge on its tail. And that would be how you'd draw the dipole between those two atoms in this bond. HCl is hydrogen chloride, and at room temperature it exists as a clear, colorless gas. You can make it, or the chlorine has a slightly negative charge, and the hydrogen has a slightly positive charge, as we saw. You can make it by reacting hydrogen gas with chlorine gas, as you can see is happening in this furnace here. And if you dissolve it in water, it becomes hydrochloric acid. Hydrochloric acid is the acid that we have in our stomachs that helps to dissolve the proteins that we eat. And what's really dissolving them are those positive and negative charges that we saw before in that dipole. How about between carbon and iodine? What's the dipole between those? So carbon is here at the top right, iodine there at the bottom right. Carbon is higher up, but iodine is further to the right. So to really tell, you'd have to go to a table of electronegativities. Carbon is 2.55, iodine is 2.66. Because iodine has a higher number, it is more electronegative, so of these, iodine would be the more electronegative element. The more electronegative element pulls electrons to itself more, and so becomes slightly negative, and the atom it's bonded to is slightly positive. You could also draw that dipole by having an arrow pointed toward the more electronegative atom with a positive charge on its tail. And so that would be how you would draw the dipole on that bond, on each of those atoms in that bond. Where would you find such a bond? You would find it in the hormone thyroxine, which is a pretty big molecule here, but it sets your metabolic rate. It sets the metabolic rate in humans so that the higher concentration you have of thyroxine, the faster your metabolism, and the lower the concentration, the slower your metabolism. And you can see that there are lots of carbon to iodine bonds here. On each of those bonds, that iodine would have a slight negative charge and the carbon would have a slight positive charge. So again, slight negative on the iodine, slight positive on the carbon. Slight negative on the iodine, slight positive on the carbon. Slight negative on the iodine, slight positive on the carbon. And it's those sticky charges that make this hormone stick to each of the receptors in your body that decide whether your metabolism is going to go slower or faster. If you have an imbalance in the amount of thyroxine, if you have a deficiency, a very large deficiency in thyroxine, you could get what's called a goitre, which is where your thyroid hormone, which is under your chin, at the top of your neck, swells, and you can prevent getting a goitre by eating plenty of iodine. And it's the reason it's to provide iodine to create thyroxine, so that people don't get goitres, that they iodize salt. Thanks so much for your attention.